The topic of today's lecture is inflammation, and specifically you will be introduced to the mechanisms of inflammation. So by the time you complete this lecture and reviewing the material, you should be able to define inflammation, the differences between acute, chronic, localized, systemic, septic, and aseptic inflammation. You should also be able to describe the vascular changes during inflammation and describe the role of the cells of the immune system and plasma proteins in acute inflammation. So the main focus of this lecture is really the learning objective number 2 and 3. We're going to talk about chronic inflammation, so you should also be able to describe the hallmarks of chronic inflammation and describe the localized and systemic effects of inflammation. And then finally, differentiate between the outcomes of acute and chronic inflammation. Inflammation is defined as a set of protective responses to cell injury that are composed of responses of blood vessels and cells of the immune system. Now, even though you might have heard inflammation in the context of disease many times, it is really a protective response to agents offending or injuring the host. Therefore, the aims of inflammation or inflammatory responses are to completely destroy and remove the offending agent from the host, or if this cannot be done, then at least trying to reduce the burden and growth of the offending and injuring agents, as well as limit the spread of the cell and tissue injuring agents. And also, the aim is to remove the consequences of cell injury. In other words, necrotic cells and apoptotic cells that result from the inflammatory response, as well as from the invading and offending agents, that is also removed by inflammatory response. Inflammation can be either acute or chronic, where acute inflammation refers to inflammatory responses lasting a few hours to a few days, in contrast to chronic inflammation, which lasts longer than a few days. Inflammation can also be septic versus aseptic, where septic inflammation is induced by microorganisms and their products, while aseptic inflammation is induced by non-microbial or non-infectious agents. Inflammation can be localized or systemic. And localized inflammation, as the term indicates, is limited to a particular location of a body, while systemic inflammation is inflammation happening throughout the body or in various parts of the body at the same time. Now, the effects of inflammation can also be localized or systemic, and the effects of inflammatory responses are mediated by, most of the time, the involvement of inflammatory cytokines, as the inflammatory cytokines can have an effect in an autocrine, paracrine, or endocrine manner. We will begin with acute inflammation, and actually this is going to be our main topic of today's lecture. And we will only touch briefly upon chronic inflammation toward the end. As it has been already mentioned, acute inflammation is a rapid response lasting for a few hours to a few days. And there are a number of stimuli that can induce acute inflammation, one of which is infections, in which detection of microorganisms by the cells of innate immunity and activation of these cells induce inflammatory responses. Non-infectious situations such as ischemia, trauma, And physical and chemical injury also induce acute inflammation, and examples include burns, irradiation, which damages the cells, and hypoxia, which is lack of oxygen, and myocardial infarction induce acute inflammatory responses. Simple presence of foreign bodies, such as splinters, dirt, and sutures from surgery, can also induce acute inflammation. And finally, excessive and dysregulated immune responses that occur in autoimmune disorders and allergic responses induce acute inflammation. So physiological consequences of acute inflammatory responses are recruitment of leukocytes and delivering plasma proteins to the site of injury. And this physiological consequences involve vascular changes, and therefore the hallmarks of acute inflammation are pain, heat, and erythema, which means redness of the affected area due to vasodilation and increased blood flow. Also, the other hallmark is edema, which refers to swelling as a result of increased vascular permeability, leading to accumulation of fluid and proteins, as well as cells in the tissue. We're now going to take a look in detail what cellular and physiological events occur during acute inflammation. So here is the list of sequence of responses during acute inflammation, which is initiated by various stimuli, and that causes changes in vascular flow and vascular characteristics, as well as changes in the lymphatic vessels and increased lymph flow, followed by leukocyte recruitment. And these recruited leukocytes then detect and remove the offending agents. And as the offending agents are removed, then the response is terminated. So over the next major segment of this lecture, we're going to take a look at each event. Now, you already know that infection, various microorganisms, as well as any other physical trauma and presence of foreign bodies can induce cell or tissue injury. And as these offending agents enter into the body of the host, they are recognized by tissue resident macrophages, as well as a number of other cells of innate immunity. Now, the resident macrophages recognize these offending agents through pattern recognition receptor binding to either PAMPs coming from pathogen or DAMPs present in damaged cells. As a result of activation of resident macrophages, then macrophages mediate phagocytosis and begin intracellular mechanisms of killing, which involves production of reactive oxygen and nitrogen species, as well as number of different pro-inflammatory cytokines, such as TNF, IL-1, IL-6, and chemokines, for example, interleukin-8. Now, in the tissue, dendritic cells are also present, and dendritic cells pick up the offending agents for protein antigens, then they process that protein antigen and they begin migrate down to draining lymph node to present that antigen to naive T cells. As you already know, mast cells are also present in the tissue, so resident mast cells can be activated by physical trauma or various subunits, various soluble components, such as subunits of complement, some microbial products, and neuropeptides that are being produced during the response. So mast cells, when they're activated in this way, release their granular content, which contains, for example, biological or vasoactive amines such as histamine, some lipid mediators, for example, leukotrienes, and various enzymes. And mast cells also produce cytokines such as interleukin-4, as well as chemokines that we have mentioned before. So here is a quick review of what is going on in tissue macrophages when there is an offending agent. That offending agents are recognized by pattern recognition receptors expressed by macrophages and taken inside a cell in the phagosome, which then fuses together with the lysosome. So at this point, creating phagolysosome leads to oxygen-independent killing mediated by lysosomal enzymes. At the same time, there is activation of oxygen-dependent killing mediated by the enzymes present in the phagolysosome membrane. That is, NADPH oxidase converts oxygen to reactive oxygen species, and nitric oxide synthase converts nitrogen to reactive nitrogen species. Now, for acute inflammation, I want to emphasize the importance of the function of nitric oxide synthase and creation of nitric oxide, which is one of the components contributing to vascular changes. So, the changes that occur in vascular flow and vascular characteristics are listed here. So it involves vasodilation and increased vascular permeability, and then a phenomenon called stasis and leukocyte accumulation along the vascular endothelium. So these are the main characteristics that occur during acute inflammation, and we're going to take a look at the mechanism of each of these. So as I have mentioned earlier, that vasoactive amines such as histamine and serotonin are produced when the cells of innate immunity are activated by the offending agents. In particular, mast cells produce histamine, and this histamine together with nitric oxide, they mediate relaxation of vascular endothelial cells as well as vascular smooth muscle, and that causes vasodilation. So as you can see here in this macrophage, inducible nitric oxide synthase leads to production of nitric oxide, and this nitric oxide acts on endothelial cells to relax. Okay, but nitric oxide synthase is not only present in macrophages as inducible nitric oxide synthase, they are also present in endothelium, and so there is endothelial nitric oxide synthase, as well as in the neurons, as in the form of neuronal nitric oxide synthase. So basically, there are different sources of nitric oxide synthase that contribute to the relaxation of endothelial cells and vascular smooth muscle transiently. This is a temporary, so transiently allowing the cell, the vasculature to dilate and allowing increased blood flow. Now, before we move on to the next change in the characteristics of the vasculature, we have to define a few terms that you need to know. The first one is edema, and edema refers to swelling caused by excessive fluids in the interstitial tissues and serous cavities. And these fluids are termed exudate or transudate. So in this figure, we see a normal situation as well as a couple of the situations where transudate and exudate is found. Under the normal circumstances, that there is good balance or physiological balance between hydrostatic pressure and osmotic pressure surrounding the blood vessel in such a way that the plasma proteins as well as other blood constituents are well kept within the blood vessel, separated from the tissue. In another situation where there is a change in these hydrostatic and osmotic pressure, then there is a leakage of the fluid from the blood vessel into the tissue through the gap junction between vascular endothelial cells. In this scenario, there is no change in the vascular endothelial cells, but because of the changes in the pressure, so for example, increase in hydrostatic pressure or decrease in osmotic pressure, the fluid of the blood seeps through vascular, through the in-between of the vascular endothelium and is collected in the tissue. The other situation is what we are discussing today is in inflammation, where there is changes in the vasculature with vasodilation and vascular permeability. And as a result of vascular permeability, there is increase in interendothelial spaces, creating the larger space between vascular endothelial cells, allowing the constituents of the blood, including fluid, protein, and cells, that they leak through the increased space and collect in the tissues. When this happens, this collection of fluid and proteins, they are called exudate. So this is just to summarize what I've just said, that transudate, so let's define it again. Transudate is fluid with low protein content, so they are mostly fluid. And when they contain protein, then it is mostly albumin. And there is little or no cellular debris, and it has low specific gravity. And it is as a result of changes in osmotic or hydrostatic pressure, causing the imbalance across the blood vessel wall without the changes in vascular endothelial cells. Now, when we're talking about vascular permeability during acute inflammation, we are basically referring to the changes in such a way that there is exudate buildup in the tissue. In other words, exudate, which is defined as extravascular fluid, which is high in protein concentration, which contains significant amount of cellular debris, and thus it has high specific gravity. And it is due to increased vascular permeability as a result of increase in interendothelial spaces. And one specific exudate that happens during infection is called pus. And pus is an exudate containing mostly neutrophils and dead cell debris, as well as components of microbes, microorganisms, and microbial products. So in the next couple of slides, we're going to take a look at the mechanisms that causes increase in interendothelial space. Okay, so at the top here, we are seeing again under the normal circumstance where vascular endothelium is nicely lined up with the tight junction gap filled and therefore separating the blood components from the tissue. The first mechanism of increasing vascular permeability is due to retraction or constriction of vascular endothelial cells. In other words, vascular endothelial cells contract and therefore it creates increased space. And this contraction or retraction is induced by histamine and nitric oxide and other mediators. And this contraction is very rapid and short-lived within ranges of minutes, and it occurs mainly in the venules. Okay, now we have seen that nitric oxide and histamine induces relaxation of vascular endothelial cells in vasodilation. But I also mentioned that that's also very transient, it's short-lived. And so similarly, vascular constriction or retraction is short-lived, ranging minutes. But nonetheless, that it causes the interendothelial space, allowing the constituents of the blood to seep through and accumulate in the tissue. The second mechanism is due to endothelial injury. Vascular endothelial cells are damaged, and this happens in the context where there is physical damage by events such as burns or infection, in which some of the microbial toxins can cause direct injury to vascular endothelium. So as the vascular endothelial cells die or they're damaged, undergoing repair process, the interendothelial space is increased, and therefore the fluid and proteins and cells migrate from blood into the tissue. And this happens quite rapidly, but lasts longer, ranging from hours to days, and it occurs in arterioles, capillaries, and venules. The third mechanism of increasing in vascular permeability is again due to vascular injury, but this time it is mediated by the leukocytes instead of the foreign entity. And it happens usually in the late stage of inflammation. So in this figure, we see now that there are a lot of accumulation or recruitment of neutrophils to the site of inflammation. And as the neutrophils are accumulated at the vascular endothelium and attach, and they're activated, as they're activated, they release their granular content and their enzymes. And due to the granular contents and enzymes that are toxic to the vascular endothelium, therefore, the vascular endothelial cells undergo death and damages. And this effect is quite long-lived, within ranges of hours, and this type of damage occurs in venules and in the pulmonary capillaries. Now, the last mechanism happens without vasculature changes and without increasing intervascular endothelial spaces, and that is called increased transcytosis. And in this situation, it does not involve cells, but it involves fluid and proteins, in that they can migrate through vascular endothelium, through transcytosis, into the tissues. And it is induced by a number of soluble factors. For example, soluble factor or cytokines such as VEGF. Now then, as a result of vasodilation and increased vascular permeability, a phenomenon called stasis occurs. Stasis is defined as vascular congestion in small blood vessels, producing localized redness, which is erythema, and accumulation of leukocytes along the vascular endothelium. And predominant population of the leukocytes are neutrophils. So how does this occur? In other words, how do vasodilation and increased vascular permeability lead to a condition called stasis? When vasodilation occurs, there is a slowing down of the blood flow due to the increase in blood diameter. With increased vascular permeability, there is loss of fluid. So together with the slowing down of the blood flow and loss of fluid lead to increased concentration of red blood cells in small vessels, and therefore increase in viscosity of the blood. Now, as the viscosity increases and blood flow slows down, the red blood cells are generally packed in the center of the small vessels, pushing the leukocytes along the vascular endothelium. So in this figure at the bottom shows a section of the small vessel where you see the vascular endothelium on the top and the bottom. And this is intravascular, so inside the small vessel. And the part that you don't see on this on this slide is the tissue part. But you can see that there are red blood cells, all congested, stained with red, here represented as red, while leukocytes are pushed against the vascular endothelium. Okay, so you see these black arrow shows the dark purple cells stained with dark purple are the leukocytes, mainly neutrophils, all along the vascular endothelium. And then you can even see the cells right here in the blue arrow. I'm going to use this blue arrow that they're right together with the vascular endothelium. Now it's trying to cross the vascular endothelium, and then now you see a couple of the cells that have crossed vascular endothelium and now on the tissue side. So this congestion of red blood cells and pushing the neutrophils or leukocytes along the vascular endothelium is called stasis. So far, we've looked at the vascular changes in acute inflammation involving vasodilation and increased vascular permeability, leading to stasis. Now we're going to move on to the recruitment and migration of leukocytes from blood into the tissue. And in order to do that, we need to go back and look at the initiation of acute inflammatory response. And that is, for example, in this figure, starting with activated macrophages under infection scenario. So activated macrophages would produce cytokines, pro-inflammatory cytokines such as TNF and IL-1. And these cytokines act on vascular endothelial cells to increase the expression of cell adhesion molecules that are selectin molecules. In the blood, flowing leukocytes would have their cell adhesion molecules as well as the ligands for selectin molecules. And so with the increase in selectin in vascular endothelium, now the leukocytes would begin to bind to the cell adhesion molecule. However, this interaction is a weaker interaction, and therefore the cells would bind and come off, roll down, and then bind again, come off, and roll down. So they would begin rolling. So we have the rolling here due to the interaction with the cell adhesion molecules expressed on the endothelial cells. Now we also know during the acute inflammation, chemokines are produced, and one of the effects of the chemokines is to act on the cell adhesion molecules expressed on vascular endothelium as well as on the leukocytes to increase their affinity. So as the leukocytes roll on the vascular endothelium, under the influence of chemokines, the affinity of another type of adhesion molecules called integrin expressed on the leukocytes would increase in its affinity and begin to bind to the ligand of integrin ligand expressed by vascular endothelial cells. As a result, together with the interaction of selectin molecules and their ligand, and then integrin and integrin ligands, now the leukocytes firmly attach to vascular endothelium, and then eventually they migrate through the space between vascular endothelial cells, come into the tissue where the acute inflammation is taking place. So here is the summary of vascular changes in acute inflammation and the immediate outcome of the changes. In comparison to normal scenario, which in this diagram shows arteriole, venule, and the capillary beds, that in inflamed situation or in acute inflammation, there is vasodilation, both in arteriole and venule, and expansion of capillary beds, increase in vascular permeability leading to leakage of plasma proteins as well as fluid. And due to stasis and leukocyte binding to the vascular endothelium, then the leukocytes, mainly neutrophils, migrate out of vascular from the blood vessel into the tissue. Now we have to remember that while the blood vessels undergo these changes during acute inflammation, that there are also changes taking place in lymphatic vessels. Now, to just review the normal functions of lymphatic vessels is that first that they are involved in draining extracellular fluids seeping out of the vascular capillaries, and so they drain from those fluids. They also traffic leukocytes throughout the body, and you know that the cells from one lymph node to the next lymph node, that they migrate through lymphatic vessels. And one important function of lymphatic vessels is that they actually drain cellular debris, microorganisms, and microbial product along with antigen-presenting cells to draining lymph node to initiate adaptive immune response. Now, during inflammation, what happens is that because of the extracellular fluid increases, lymphatic vessels proliferate to accommodate the increased volume of fluids coming out of the blood vessels. And this may lead to secondary inflammation in lymphatic vessels. And when that happens, it is called lymphangitis. And as the cells and cellular debris and microorganisms and microbial products are drained into the draining lymph nodes, there is extensive activation and proliferation of lymphocytes and increased number of macrophages present in the draining lymph nodes, leading to enlargement of lymph nodes. And sometimes it can turn into reactive or inflammatory lymphadenitis. So this slide is just to simply remind us that during acute inflammation, the changes occur not only in blood vessels, but also in lymphatic vessels. And this graph shows the time course of the responses that occur during acute inflammation, looking at edema and cellular infiltration at the site of inflammation. So here on the x-axis, we're looking at number of days, and this is intensity of the responses or activity, showing that edema occurs first within 1 day, and then cellular infiltration of predominantly neutrophils occurs first, and then from blood, monocyte infiltration takes a couple days to build up to reach maximum, and then they linger on at the site of inflammation a little longer than neutrophils and fluid. So as the result of the accumulation of the cells at the site of inflammation, then these cells are involved in removal of the offending agents that initiated the inflammatory response. And in the tissue, you already know that the macrophages are present and neutrophils are recruited to the site. Together, there are phagocytes, and they mediate phagocytosis and reactive oxygen species, as well as pro-inflammatory cytokine production. Now, I want to just add, in addition, for neutrophils that they can release lysosomal enzymes to remove the offending agents. Similarly, granulocytes that are found in the tissue are mast cells, and they can be activated and they can release granular content in acute inflammation as well as in chronic inflammation. They themselves can produce a variety of cytokines, but these cytokines can either promote or limit inflammation depending on the situation. Regardless, the classical example of cytokines that mast cells produce are interleukin-4 and the chemokine interleukin-8. In addition to neutrophils, during inflammation, eosinophils can be recruited by chemokine, and specific chemokine that recruits eosinophils is eotaxin. And this is particularly important in parasitic infection. And in this case, they release their granular content containing major basic protein, which is toxic to the parasite, but it can be toxic to surrounding host cells as well. The next couple of slides, I have summarized the mediators involved in acute inflammation. This slide groups them in terms of their classes. So like cytokines and chemokines, plasma proteins, vasoactive amines, etc. But these soluble mediators have been mentioned in the process of describing acute inflammation, so I will not go over them again, other than the fact that I want to highlight that you need to know acute phase proteins and that the fact that they are involved in acute inflammation, and also neuropeptides released by sensory nerves are involved in acute inflammation, and major examples are substance P and neurokinin. This slide has a table that grouped the soluble mediators in terms of their function, in terms of their role in inflammation. So, for example, in vasodilation, you can see different classes of soluble mediators contributing to vasodilation. All right, so this is not for you to memorize every single one of the mediators, but that you would know that there are a number of different soluble mediators contributing to different aspects of inflammation, even though they may not have been mentioned during the lecture. As the offending agents are being removed, then the responses in acute inflammation need to be downregulated and eventually terminated. The termination process is mediated by natural degradation of inflammatory mediators. These mediators have short half-life, and therefore they degrade shortly after their release. The other thing is that the activated neutrophils that migrate from blood to the tissue also have short half-life, and therefore they only survive for a few hours after leaving blood. Second way of terminating the response is inducing stop signals actively. And these signals are triggered by production of anti-inflammatory cytokines such as TGF-beta, tumor growth factor beta, and interleukin-10 by a subset of macrophages, and there are other cells that can produce these anti-inflammatory cytokines. Also, production of anti-inflammatory lipid mediators induce active signal to downregulate and terminate inflammatory response. And major examples of these lipid mediators are resolvin and protectin, and they're derived from saturated fatty acids. Another way to induce stop signal for inflammatory response is converting arachidonic acid metabolites from pro-inflammatory metabolite into anti-inflammatory metabolite. So in inducing inflammation, arachidonic acids are converted to leukotrienes. However, in the phase of terminating the response, arachidonic acids are converted to anti-inflammatory metabolite such as lipoxins. Last but not least is inhibition of pro-inflammatory cytokine, TNF, production in macrophages by neural impulses. And these neural impulses are mediated by cholinergic discharge. Before we close the segment on acute inflammation, I would like to highlight the effects of inflammatory cytokines, TNF and IL-1, and the same principle also applies to interleukin-6. We know that TNF and IL-1 are produced by activated macrophages during acute inflammation when these cells are activated, and their effects can be local or systemic. Local effects of TNF and IL-1 are on the vascular endothelium and leukocytes, and as we have seen in the majority of this lecture, that these effects induce inflammatory responses. The local effects can also be on fibroblasts in the affected area, in which it promotes, or these cytokines promote proliferation of fibroblasts, as well as increase in collagen synthesis. And this promotes tissue repair. The systemic effects of TNF and IL-1 include fever, increased production of leukocytes in the bone marrow, and increased production of acute phase proteins in the liver, as well as other symptoms such as decrease in appetite and increase in sleep. These systemic manifestations of inflammation they are observed even when the acute inflammation is localized. Here are the different possible outcomes of acute inflammation. First, it may be completely resolved and the affected tissue regain the normal function. It happens when there are effective clearance of injurious stimuli or offending agent, and also clearance of mediators of inflammation as well as inflammatory cells, and the injured cells are replaced with normally functioning tissue cells. And as a result, the normal function of the area or normal function of the tissue is regained. The second possible outcome is progression to chronic inflammation. And it happens when the clearance of injurious stimuli or offending agents is not successful and they persist in the host. Third outcome may be healing of the injury. That is, effective clearance of the offending agent and inflammatory mediators do occur, but the injured cells are replaced with scar formation. In other words, replaced with fibrotic cells and connective tissues instead of normally functioning tissue cells. So then final outcome is while the affected area is healed, it has been healed with loss of function, and that is called fibrosis. The last outcome may be occurring as a result of microbial infection, in particular bacterial infection, where there is a severe infection leading to pus formation or, in other words, abscess formation. So when abscesses are effectively cleared and the area is healed, this healing again involves the replacement of injured cells, injured area with fibrotic cells and connective tissues, and as a result, while the microorganisms have been cleared, the affected area is not regaining the normal function, but rather it is replaced with fibrosis. In the end, ultimately, either acute inflammation is either completely resolved or it turns into fibrosis. Now, in the remainder time of this lecture, we will discuss chronic inflammation. And I will just point out the major features of chronic inflammation, and we will not discuss the mechanism of chronic inflammation, as the mechanisms tend to be more varying and more complex. Chronic inflammation is defined as inflammation of prolonged duration ranging from weeks to months or sometimes even years. And it is due to varying combinations of inflammatory responses in which tissue injury and tissue repair attempts may coexist. In other words, there is tissue injury and then tissue repair. While tissue is being repaired, there's another set of tissue injury, etc. Chronic inflammation may occur as a follow-up response to an acute inflammation, or it may occur insidiously, which means that it is occurring in a gradual and subtle way as a low-grade response without physiological manifestations of an acute inflammatory response. Now, this insidious response is the primary cause of tissue damage in many of the human diseases. For example, rheumatoid arthritis, which is an autoimmune disorder, or atherosclerosis, or microbial infection leading to tuberculosis, and pulmonary fibrosis. Now, there are different causes that can induce chronic inflammation, and one is persistent infection by microorganisms, such as mycobacteria, but it can also happen with certain viruses, fungi, and parasites when they have persistent infection. Second cause is immune-mediated response, where the immune response is excessive and inappropriate and not well regulated, as we see in autoimmune disorders and allergic responses. Also, prolonged exposure to toxic agents can lead to chronic inflammation. And these toxic agents are non-biological agents, and it can be exogenous agents and endogenous agents. Exogenous agents are particulate silica that may be found in the environment, and prolonged inhalation of these silica can induce inflammatory lung disease. Endogenous agents include toxic plasma lipid components such as low-density cholesterol, inducing chronic inflammation of arterial wall leading to atherosclerosis. Now, chronic inflammation is characterized by these three main characteristics. That is, first, infiltration with mononuclear cells at the site of inflammation. And these mononuclear cells are macrophages and lymphocytes, both T and B lymphocytes as well as plasma cells, instead of neutrophils and monocytes in case of acute inflammation. Secondly, it is marked by tissue destruction mediated by injuring agent or offending agent persistently present in the host, and as a result, inflammatory cells are also persistently present at the site of inflammation. Third, chronic inflammation is marked by replacement of damaged tissues with connective tissue. And this is a result of a progressively irreversible fibrotic response in which accumulation of components of extracellular matrix such as collagen and fibronectin in and around the injured tissue. Therefore, the tissue repair occurs with non-functioning components of extracellular matrix rather than functioning tissue cells. And this fibrosis is accompanied with angiogenesis, which is generation of new blood vessels, as well as growth of new lymphatic vessels. And here is the summary of chronic inflammation that we have discussed in this lecture, including the outcome. And we have looked at a number of different types of injury that would induce chronic inflammation. And that in chronic inflammation, a mononuclear cell infiltration, angiogenesis, and fibrosis are observed. And then there will be continuous cell injury and attempts to repair would be present. Therefore, the final outcome of chronic inflammation is fibrosis with a significant amount of collagen deposition or fibronectin deposition in and around the inflammatory site, and rather than replacement with the functioning cells, and therefore it leads to loss of the function of the tissue being affected. So that is the end of today's lecture on inflammation. And if you have any questions, please do bring them to tutorial sessions we will have next week.